at a raw potato and a block of wood. If you strip away the macroscopic textures and zoom down to their molecular foundation, you will find the exact same repeating chemical structure. Yet, if you eat the potato, your body will readily extract enough metabolic energy to keep you alive. If you try to eat the wood, you will starve to death, unable to digest a single calorie of it. Both the starch inside the potato and the cellulose that makes up the wood are polymers built entirely from identical monomeric units, D-glucose, a simple sugar with the formula C6H12O6. We cannot point to the atoms to explain why one material is a fuel source and the other is an impenetrable building material. The carbon, hydrogen, and oxygen counts are exactly the same. The difference lies entirely in the three-dimensional geometry. In biological systems, a molecule's physical function is dictated by the precise spatial arrangement of its atoms at the nanometer scale, its stereochemistry. By analyzing the structural logic of biomolecules, carbohydrates, proteins, nucleic acids, and lipids, we can reverse engineer how a minor shift in a single chemical bond translates into the macroscopic physical properties of living organisms. This diagram shows the linear Fischer projection of open-chain D-glucose. In an aqueous environment, this chain folds to form a six-membered Haworth-Pyranose ring. The ring closes via a specific mechanism. The C5 hydroxyl group executes a nucleophilic attack on the C1 carbonyl group. This reaction locks the molecule into a ring, generating a new chiral center at carbon 1, the anomeric carbon. This closure yields two distinct geometric isomers, alpha D-glucose with an axial downward hydroxyl, and beta D-glucose, where it points equatorially up. String thousands of alpha D-glucose molecules together via alpha-1,4 glycosidic linkages, and you get amylose, the primary component of starch. The repeating downward geometry of those alpha bonds physically forces the polymer chain to curve. It coils into a right-handed helix. This open, helical structure is highly soluble in water and perfectly fits the active site of human amylase enzymes, allowing us to break it down for energy. Now look at cellulose. It uses the exact same D-glucose monomers, but connects them using beta-1,4 glycosidic linkages. Because that beta hydroxyl group points out equatorially, the resulting polymer does not coil. The chains lie perfectly flat. This linear geometry allows parallel sheets of cellulose to stack flush against each other, locking together through extensive, highly stable hydrogen bonding. Mammalian digestive systems lack the specific cellulase enzymes required to chemically cleave that beta-oriented bond. The sheets remain locked tightly together as they pass entirely through the gut. The mere difference of a hydroxyl group pointing down or up is the sole chemical distinction between a potato and a piece of wood. This principle of geometry dictating physical properties extends directly to structural proteins. Every protein begins with the alpha amino acid, a bifunctional molecule containing a basic amine group on one side and an acidic carboxyl group on the other. In a neutral solution, the acidic carboxyl group donates its proton to the basic amine group. The molecule now carries both a negative and a positive charge, becoming an internal salt known as a zwitterion. Every amino acid has an isoelectric point, the specific pH where the zwitterion carries no net electrical charge. Placed in an electric field at this pH, the molecule remains completely stationary, migrating toward neither the cathode nor the anode. To build a protein, the carboxyl group of one amino acid links to the amine group of the next. This condensation reaction eliminates a molecule of water and forms a strong covalent peptide bond. The resulting primary sequence of amino acids is highly flexible. Because there is free rotation around the single bonds within the polypeptide skeleton, the chain can fold into a nearly infinite number of shapes. But it doesn't. The chains collapse into fixed, rigid secondary structures, driven by hydrogen bonding. The two most common are the alpha helix and beta-pleated sheet. When the peptide chain twists into a right-handed coil, it forms an alpha helix. These hydrogen bonds allow the molecule to act like a physical spring. This molecular elasticity is exactly why proteins like keratin make your hair stretchable. Alternatively, multiple peptide chains can align side by side. Hydrogen bonds lock the adjacent chains into a flat, rigid grid called a beta-pleated sheet. This specific geometry gives materials like silk their high tensile strength and lack of stretch. The macroscopic texture of biological tissue, whether stretching and snapping back or holding firm, is a scalable consequence of nanoscale hydrogen bonds locking peptide sequences in place. This geometric precision is most critical when a living system needs to store information. The modular unit of genetic data is the nucleotide, which consists of a phosphate group, a pentose sugar, and a nitrogenous base. The diagram here shows the pentose sugars for RNA and DNA. They are identical except for the specific substituent at the 2' carbon. Ribose has a hydroxyl group here, while deoxyribose has a simple hydrogen atom. Lacking that 2' oxygen atom restricts the molecule's chemical reactivity. This seemingly minor omission provides DNA with the immense chemical stability required for long-term genetic archiving. The stability of the double helix relies on strict geometric complementarity. The purine base adenine always pairs with the pyrimidine thymine, interlocking perfectly via exactly 2 hydrogen bonds. Likewise, guanine pairs exclusively with cytosine, locking together with 3 hydrogen bonds. By always pairing a double-ring purine with a single-ring pyrimidine, the DNA double helix maintains a rigid, uniform width of exactly 20 angstroms from end to end. Mapping these rigid spatial constraints required building actual physical models to test which molecular geometries were mathematically possible. Establishing the anti-parallel, complementary nature of DNA was the result of a massive multidisciplinary effort to map physical chemistry to biological inheritance. And that structural breakthrough paved the way to decode the genetic language entirely, mapping exactly how nucleotide triplets physically translate into the amino acids building proteins. We see this exact principle of geometric assembly in the cell membrane. A phospholipid is composed of a polar, hydrophilic head containing a phosphate group and 2 nonpolar, hydrophobic fatty acid tails. When placed in the aqueous environment of a living cell, this amphipathic geometry forces a thermodynamic reaction. To minimize contact with water, the hydrophobic tails point inward toward each other, while the hydrophilic heads face outward. The lipids spontaneously self-assemble into a continuous double layer. Biological assembly requires absolutely no outside architect or physical interference. Whether locking genetic code into a double helix or walling off a cell membrane, the geometry of monomers automatically drives the construction of complex systems. There is one hurdle remaining. The process of joining these highly ordered biological polymers together, anabolism, requires a massive input of energy. The universal energy currency that traps, stores, and transfers this required power is adenosine triphosphate, or ATP. Look at this diagram of the ATP molecule's triphosphate tail. It consists of a sequence of 3 phosphate groups attached to a ribose sugar. Notice the 4 negatively charged oxygen atoms packed tightly together along this chain. These identical charges experience severe electrostatic repulsion. The molecule is essentially a loaded molecular spring, highly unstable and desperate to push itself apart. When water breaks the terminal bond, hydrolyzing ATP into ADP and an inorganic phosphate, that electrostatic tension is relieved. The cleavage of that specific bond releases exactly 30.93 kilojoules of energy per mole. This precise packet of chemical energy is what living cells use to drive the dehydration synthesis reactions. It pays the thermodynamic toll required to assemble the starches, fold the proteins, and synthesize the DNA. Biological life is a manifestation of applied organic chemistry. Every capability of a living organism, from metabolic energy transfer to structural integrity and genetic inheritance, is strictly dictated by nanometer-scale geometry, precise stereochemistry, and the inevitable flow of thermodynamic gradients.